Right, so for this particular part of the video, what we are going to be looking at is the function of the myelin sheath in transmitting action potential or nerve impulse. Now, just a bit of revision. We've covered the myelin sheath before in a previous video where we talked about the structure, and I told you that the myelin sheath are just these particular structures made out of something called the Schwann cell, and the Schwann cell wraps around the axon to form the covering of the myelin sheath, and certain parts of the axon which are not myelinated are referred to as something called the nodes of Ranvier. Now I'm just drawing it back over here, and as you can see here, certain parts are myelinated and certain parts are not myelinated. If we were to magnify, if we were to just zoom in a little bit here, I want you to see the concentration or the amount of sodium ion channels along the axon. Again, we are just focusing on the sodium ion channels. We are not focusing on the sodium ion potassium ion pump or the potassium ion channels, not because they don't exist. They do exist, but the sodium ion channels are just the core focus over here. So for this part, what I want you to see is, I want you to notice that for the part of the axon where it's myelinated, you notice that there are less channels or less sodium ion channels, and for the nodes of Ranvier, which are not myelinated, they have more sodium ion channels. And also, by the way, the myelin sheath provides an area of electrical insulation. What I mean by electrical insulation is the ions outside the axon will have great difficulty trying to diffuse into the axon because the myelin layer is made up of predominantly lipids. So lipids are mostly nonpolar. I'm not saying that phospholipids can be polar as well, but the lipids are predominantly nonpolar, so they do repel the ions away. And even if they don't repel the ions away, the lack of channels make it difficult for the ions to either diffuse into the axon or diffuse out of the axon. But conversely, in the nodes of Ranvier, because they don't have those insulation and they have more channels, the ions will have an easier time diffusing into the axon or diffusing out of the axon, depending on the concentration gradient. So you might be thinking, okay, big whoop, why do I care? So let me just write it back again. The myelin sheath provides an area of electrical insulation, so therefore the ions cannot pass through, and the nodes of Ranvier have no electrical insulation, so ions can pass through. Some students will say, oh, parts of the axon which are myelinated or have the myelin sheath are less permeable to ions, and the nodes of Ranvier are more permeable to ions. That is a valid statement that you can also make. So what's the big deal? Why do we have to care about this insulation or such? The reason is because the function of the myelin sheath is to speed up the action potential. Now, some students will immediately go, how does that happen? Because I just mentioned that the myelin sheath prevents ions from passing through. So some students will say, shouldn't it actually reduce the action potentials, right? It shouldn't speed up the action potentials. So let's look at it. Now, what I'm drawing over here is the axon. I'm drawing out two different axons, by the way. It's not a time lapse, okay? Two separate axons. And the axon at the top is unmyelinated and the axon at the bottom is myelinated. As you can see, the axon at the bottom has those myelin sheaths, just those parts where I've highlighted in beige. Is that beige? Beige, maroon? No, not maroon, definitely not maroon. Beige or brown, peach, whatever. Okay, right. So let's look at the unmyelinated axon. Now, for the unmyelinated axon, if a stimulus is given to that section of the axon, what happens is the first part of the axon will undergo depolarization where sodium ions diffuse into the axon, so the membrane fully depolarizes, assuming that it reaches the threshold potential, by the way. And when one section depolarizes, it produces a local circuit. Local circuit will cause the next section's sodium ion channels to open and sodium ions rush in, and therefore that next section will also depolarize. Again, It produces another local circuit which causes the next section sodium ion channels to open, so sodium ion rushes in, which causes the next section to also depolarize, and repeat ad nauseum until all the way towards the end of the axon. We've studied this in the previous chapter, where local circuits produced in the axon will cause the next section to depolarize, which causes the next section to depolarize, and so on and so forth. Okay, fine. So we've covered that. That's normal stuff. But let's look at the myelinated axon. So I'm just going to zoom in a little bit over here. When we give the stimulus to the myelinated axon, the first part, same thing. The sodium ion channels open. So sodium ion rushes in. The membrane depolarizes. So far, so good. Everything is the same. But the problem here is local circuits that are produced, which means the sodium ions inside the axon, they will diffuse into the next section. But when they diffuse into the next section, they are supposed to stimulate the sodium ion channels in the section that I'm circling over there. But I told you in this part of the video, I said that in the myelinated part of the axon, there are a lack of channels. They may have some channels, but it's very few. It's almost insignificant. And even if the channels open, sodium ions cannot diffuse in. The reason why is because the myelin sheath provides an electrical insulation. So that part of the membrane doesn't depolarize, but the local circuit will just move forward, which causes that section of the axon, which has a lot of sodium ion channels, as I'm drawing out here, and those sodium ion channels will open, which causes sodium ions to rush in, and therefore the membrane will depolarize. So, as you can see over here, the myelin sheath provides an area of electrical insulation, and therefore this causes the action potential to jump along the nodes of run VA. Now, you might be thinking, why? Who's jumping? What is jumping? There's no actual jumping going on. If you look at the axon at the top, when the first section depolarized, as I'm just putting it in a green circle, it causes the section adjacent to it or directly next to it to also depolarize. So one, this depolarizes, and then next one depolarizes. But for the axon at the bottom, however, when the first section depolarizes, as I'm circling in green, it causes the next section, which is not directly next to it. It's like it looks like it's jumping. So first it happened over here, and then it happened over there. So it implies that the action potential or the depolarization is jumping along the nodes of run VA, and therefore this will actually speed up the action potential. And by the way, we also call this the saltatory conduction, because the saltatory conduction just implies that the action potential seems to be jumping. It's the word jump. The word jumping here is used in a figurative sense. No one is jumping, by the way. Okay, nothing is jumping. Some students are like, teacher, is the axon actually jumping? No, it's not. Okay, right. If you still don't know how this speeds up the action potential, don't worry. Let's look at it again. I'm going to draw out the axon. Axon at the top is unmyelinated. Axon at the bottom is myelinated, as you can see over there. Now, let's assume that we stimulate both the axons at the same time. Okay, stimulate both the axons at the same time, as you can see here. So what happens? Both sections of the axon depolarizes. How does it depolarize? Because sodium ions rush in. Fine. Now, this produces a local circuit, but look at the local circuit. Okay, the local circuits produced will depolarize the second section, right? But for the axon at the top, it depolarizes directly the next section, okay, the section directly next to it. But for the axon at the bottom, at the same amount of time, it jumps and it skips the myelinated part, and it depolarizes the unmyelinated area, which is the node of run VA. Let's try again. What happens then? Local circuit, depolarization. Again, local circuit, depolarization. Local circuit, depolarization, local circuit, depolarization. So look at the nerve impulse. At the same amount of time that has passed, for the unmyelinated part, they are just halfway through the axon. But for the myelinated axon, the action potential has already reached the end at the same amount of time. That is how myelin sheath actually speeds up the action potential. All right, as you can see here, I'm just drawing out the arrow. The orange color arrows imply that the depolarization takes time to happen. It takes one section followed by the other section followed by the other section directly next to it for the unmyelinated axon. But for the myelinated axon, the depolarization seems to be jumping or leaping along the nodes of Ranvier, and that is known as the saltatory conduction. So if a question were to ask you what exactly is the function of the myelin sheath, all you have to say is the myelin sheath provides an area of electrical insulation which causes the action potentials to jump along the nodes of Ranvier. And by jumping along the nodes of Ranvier, this will actually speed up the action potential. That's basically it.